Okay, a couple things here before we get started. Of course, next week, Wednesday and Friday, we have guest lectures. Monday, I'll be talking about the single-stranded DNA viruses of bacteria. Wednesday, my graduate student will be talking about some of the single-stranded DNA viruses of eukaryotes, and then this new single-stranded DNA virus, which we found in a volcanic hot spring. On Friday, I'll be in my best robes, carrying my hat in my left hand as I come in for the defense of PhD thesis. Pardon?
Don't forget the tassel out.
The tassel out, exactly. Tassel down. And then the discussion mostly that we're going to be having actually has to do with some of the stuff we talked about in terms of virus taxonomy a couple of lectures ago, because they think they found an example of a bacterial virus that's actually really similar to a lot of the eukaryotic viruses, and also not unlike the T equals 31 STIV particle structure in terms of the capsid proteins. I'm not sure I completely believe them, so it'll be an interesting discussion in terms of that. So that's next week. The following week, Monday will be review, and then Wednesday is the midterm already. How did that happen? I have no idea. Other thing, hopefully most of you know OMSI. If you don't, you shouldn't. There's going to be a new exhibit there, probably about the end of the term, called Nanoland. And the idea is when you go into it, you get shrunk down to the nanoscale. And what happened was, as the people from OMSI were going through our electron microscope facility, I happened to be at the electron microscope at the time and was going, check out my new viruses, isn't this so cool? I've got tons of viruses. And they got excited enough about it that I'm actually part of the exhibit. And one of the, not physically, I'm not that small. But one of the things they're going to do after we had a couple of conversations is a hands-on kind of, how you would be acting if you're at the nanoscale. And remember, the target audience for most of the OMSI visitors is sort of between 7 and 10 years old. So this is the game, which the 7 to 10-year-olds and maybe even some of the older people might get to play there, is this. So each area has a game or action we ask visitors to do. The game instructions are trying to get across just one salient point about each world. There are multiple different nano worlds that they have there. And I'll let you guys guess what that's going to be for this case. In this case, that the viruses are trying to find their host cells so they can reproduce. Here are the game instructions. Floor instructions. As you walk in, this will be the instructions on the floor. Stop! You are a green virus. Your job is to lock onto a cell and trick it into making more viruses. You are sticky and will stick to anything. Be careful not to touch another virus. You're also bouncy. Bounce. Do you see a blue host cell? Can you bounce there without sticking to anything? You made it. Lucky you. Lock onto the cell and give it a squeeze. You get to reproduce. So I can't wait to see the kids actually being viruses. I think that's going to be hilarious. Should be a ton of fun. And I also just noticed that we're living in the past because this is from last year. But no, the questions are not from last year. The clicker questions are from this year, which are as follows. The conformational change, oops, we're going to talk here, that causes membrane fusion mediated by HA2 is due to sialic acid binding, binding to CD4, neuraminidase, a lowering of pH, and increase in pH. Yes. Talk to your neighbors, please. This is counting down, by the way. So nobody liked binding to CD4 because it's the receptor too?
HIV.
Thank you. Co-receptor, actually there are two of them, which go with that. Nobody liked neuraminidase because there's actually not a conformational change that happens in neuraminidase. It's an enzyme which actually cleaves off sialic acid from the surface of cells. We didn't talk too much about this. We'll talk more about it when we talk about influenza later on. But this is a way that the virion doesn't bind to the cells that it's already infected and is being produced by. So that neuraminidase there, and if you don't have neuraminidase, you end up getting stuck to the cells that are producing the virus, which is why blocking neuraminidase is actually quite a good drug target in terms of looking at HIV. Increase in pH, I don't know anywhere inside the cell that you get an increase in pH. So that brings us to two possibilities, sialic acid binding and a lowering of pH. Where does pH get lowered? In the endosome, moving towards the lysosome process. In fact, a lot of viruses use that, including influenza, which is HA2. You do have sialic acid binding, which causes a conformational change, but it's not a conformational change which is causing the membrane fusion. So our answer here is D. Okay, I always have to remember to close the results because it'll do, yeah.
Salicylic acid binding from HA1?
Yes, HA1. So our second clicker question, oops, that would not be this one. It would be the next one. Let's go to the next one. Here we are. Production of the lysis protein in single-stranded RNA bacteriophage MS2 is due to competition between the replicase and ribosome for binding, different secondary structures that form early and late in infection, binding the replicase protein to the genome, binding the capsid protein to the genome, or incomplete translational termination. Guessing doesn't cost you anything. 10, 5, 2, 1. Okay, quick thing about clicker questions. I was told at the beginning of the term there was this great software I could do one click from my computer up to D2L and it worked wonderfully. I've been trying to troubleshoot this for the last 4 days. So eventually we'll get the scores and everything up there so you'll know how you're doing for all of these things. So there seems to be considerably more difference of opinion on this one. Turns out that almost all of these, I can move this over to the side here real quickly, are different ways that the different amounts of proteins are regulated in MS2, but it's the correct answer is incomplete translational termination because it's the ribosome which is bouncing back and forth before dissociating from the RNA. But those of you who have to take off, have a nice weekend, enjoy it. But I did want to talk about some of the other things as well because this does have to do with regulation of the other proteins in MS2 and how those are actually being regulated. So competition between the replicase and ribosome for binding, which is number 1 up here, is regulation of which protein? So ribosome and replicase. So this is actually down-regulating the coat protein. And so once you actually make enough of the replicase protein, you want to down-regulate coat protein. Why? Because you don't want the ribosome in the way when you're actually trying to make that negative strand. Different secondary structures form early and late in infection is for not so much the lysis protein, but much more for the maturation protein at the very beginning of the genome. You only make the maturation protein if it's newly made RNA. Binding the replicase protein to the genome actually is kind of like A, so fortunately neither of them are correct. And of course, incomplete translational termination is what's happening with the lysis protein. Okay, so I think that's most of the review I wanted to do on the small RNA phage. We also talked a little bit about the biotechnology right towards the end here, and the main point about the biotechnology applications of these small RNA phage is really good RNA-dependent RNA polymerases that can produce just a ton of RNA as long as they're given the right template. And so usually it's an amplification process. Almost always this is used in a qualitative fashion, so presence-absence kind of things. Things like HIV or hepatitis C, all of these which have RNAs which it'd be nice to be able to detect and then get some kind of idea what's actually present there. Any questions more about the small RNA phage? We'll talk much more about the other RNA viruses infecting other kinds of organisms after the midterm. Okay, so today, switch things up a little bit. You may have noticed chapter-wise, we skipped over chapter 6 and went to chapter 7. That's so that we can do single-stranded DNA viruses next week. So the big picture here is Bacteriophage T7. These are double-stranded DNA bacteriophage, highly dependent on host machinery, and we'll talk about all that host machinery as well. Talk a little bit about some of the T phages like T4. This is unfortunately rather confusing because it has nothing to do with the triangulation number for icosahedral structures. So try and keep those. It's their T equals will be your triangulation number, and T 2, 3, 4, 5, 6, 7, actually went all the way through T9, are the T phages. In this particular case, and this is specific for Bacteriophage T7, and actually the really closely related Bacteriophage T3, transcription is the really important aspect about entry, how you get the genome actually inside the cell. And then one of the differences between this particular bacteriophage and some of the single-stranded DNA bacteriophage we'll be talking about next week have to do with viral polymerases. And there are viral DNA polymerases, viral RNA polymerases in the case of Bacteriophage T7. We're going to be talking a lot about regulation. We talked when we went over sort of the overall scheme of things. You've got interaction with the outside of your host cell, release of the genome, then expression of the various different genes, early and late genes, and we'll see also we're going to bring in the middle genes as a separate kind of regulatory process here. The way regulation works in Bacteriophage T7 is what people call a cascade of promoters. So you have early promoters, the products of the genes made from the early promoters will turn on the middle promoters, which will then in turn turn on the late promoters. So it's this cascade of promoters, pretty typical not just for bacteriophage replication, but also for a lot of regulation that you have in bacteria. T7 RNA polymerase, this is a pretty unique enzyme that has been incredibly useful also for biotechnology purposes that we'll talk about a little bit more later on. Probably the most unique aspect about the T7 RNA polymerase is it's a single subunit. And all of the, that should actually be T7 DNA-dependent RNA polymerase, all the other DNA-dependent RNA polymerases have lots and lots of subunits, anywhere from 5 in most bacteria to, depending on which one it is in eukaryotes, 12 or even as much as 16 different subunits that you have in the DNA-dependent RNA polymerase. It's a single subunit RNA polymerase, and it's a really good RNA polymerase as well. We already talked about early, middle, and late genes, and then how these genomes actually get finished and replicated is an interesting problem. It's the same thing as we have with all cellular replication. Whenever you start replication with a DNA-dependent DNA polymerase, you need an RNA primer. That RNA primer gets degraded, and so you end up losing pieces at the end of your genome. This is a real problem, particularly if you have a small genome like a lot of viruses do. So there has to be a way to deal with that problem. How do bacteria normally deal with that problem? Circular genomes. How do we deal with that problem? Telomeres. Exactly. The way that these virus genomes deal with it is concatamers, which is literally lots of copies of the genome, I should have them this way around, hooked up end to end to end. And many, many, particularly bacteriophage genomes, deal with this in this way. And it turns out that a number of eukaryotic viral genomes as well make these multiple copies of the genome lined up end to end. So again, outline of what we're going to talk about today. Where do these T phages come from originally? What's the structure look like? And it's actually really kind of convenient because the T7 phage actually has a T equals 7 icosahedrally symmetric major capsid. Talk a little bit about binding and entry, and later in the last couple of years we've learned a lot more about binding and entry, and I have a nice little video about that from a science paper from 2 years ago. Transcription, really a lot of what's going on in Bacteriophage T7 has to do with transcription and transcriptional regulation. Replication is actually pretty boring and easy with the exception of those concatamers. And then we'll talk about biotechnology again, mostly using the T7 RNA polymerase. So where do these type phages come from? A lot of this comes from the phage group, and those of you in the molecular genetics class have started to hear about the phage group. We're going to be hearing more and more about it. Basically, this was a group centered around Max Delbruck and Salvador Luria, who decided that bacteriophage were really the way to go to figure out life. And these were physicists. They said, hey, we want to do something nice and simple. We can use equations to solve it. How are we going to do that? We'll pick the simplest kind of life that we can think of, and that was the bacteriophage. So the problem is, at that time, which was late 40s and the early 1950s, and even to some extent a little earlier than that, was that bacteriophage were not well classified. Some people would call a bacteriophage alpha. Somebody else would call the same bacteriophage B, all kinds of different nomenclature. And so basically, Max Delbruck said, hey, guys, forget this. We have to name these different phage, and we're going to keep calling and using them exactly the same way. So that's where all of these T phages came from. And they had T1 through T9. This was also before there was much use of electron microscopy, so they actually couldn't tell what these virions actually looked like. So this is all based on the plaques. You remember the plaque assays that we talked about? Talk about one-step growth curves. How do you count the number of viruses which are there? It was just different plaque morphologies. Are they big? Are they small? Do they have a little dot in the middle? Do they not have a dot in the middle? That's where all of this came from. So it turned out much later that most of the T even, so T2, T4, T6, are really similar to each other. And the odd ones are mostly similar to each other, and particularly in the case of T3 and T7. And they're practically identical to each other with some interesting differences that we'll come back to a little bit later on. Where did they get them from? Well, the classic place that you get bacteriophage from, which is sewage, exactly, and a commercial phage preparation. Why commercial? What was this commercial aspect? Phage therapy. So these were using bacteriophage as antibiotics, which was being used, particularly in the 30s and 40s, as a way to treat bacterial disease. Lost in antibiotics, actually coming back today. And in fact, one of the people who's doing a lot of work on T7 has actually moved a lot into doing bacteriophage therapy. Why were people so excited about it, and particularly about bacteriophage T7? And this has to do with bacteriophage genetics and being able to make conditional mutants. And so the whole idea of a temperature-sensitive mutant really came up in studying the replication of bacteriophage. And it turns out you can make lots and lots and lots of bacteriophage. I showed you the tube, I think lecture 1, where you had 10 to the 11th phage particles in it. Even at very low mutation rates, you can get lots and lots of different mutations in there. So very often these were the temperature-sensitive mutants, where you have a bacteriophage that can grow normally on E. coli at one temperature, but can't replicate at a slightly different temperature. And these are really nice because you can actually study the broken form of whatever you're looking at and then figure out what's actually broken in that case. Classic case that we have for looking in genetics. And what do geneticists do? They break things and then try and figure out what they broke. So here was really very, very useful. And literally hundreds of different mutants in the Bacteriophage T7 genome. And also, it turns out they're really easy to make later on. Podoviruses, which are the class that these Bacteriophage T7 fits into, are the foot virus. And so it's foot versus tail. And so when you think about Bacteriophage T4, it's got this really long tail structure here. Basically, the podoviruses are when you don't have that long tail anymore. It's just a really short tail. Turns out they're extremely common in pathogens, and maybe even the most common virion on this planet is a podovirus. And we'll get to that towards the end of today's lecture. So again, easy to remember. T7 has a T equals 7 icosahedrally symmetric head structure. And we'll get the pen back up here so we can take a look at this. How do we get T equals 7? I'm going to do red. Sorry for those of you who like the other colors. So here we have a 5-fold axis of symmetry. Here we have a 5-fold axis of symmetry. We've got to get from here to there via the 6-fold symmetric axes. So we start here. We find a 6-fold right there. So that's one step. Now we change direction. So that was H equals 1. Now we go 1, 2, and the opposite direction. So, or orthogonal direction, any way you want to talk about it. So it's H equals 1, K equals 2. So that's 1 plus 2 plus 4 is 7. Make sense? Yes? Everybody happy? Were I to give you something like this on an exam, we could all get the right answer? Yeah? Way easier than that one I had for the clicker question last time? Yes. Okay, good. So many people will ask me, do you want us to know all of these GP numbers? No. I can't remember all these GP numbers. It's absolutely ridiculous. And this is for every given bacteriophage. And turns out every phage biologist gives their favorite bacteriophage a similar kind of name. And so GP1 in one bacteriophage could be completely different than GP1 in another bacteriophage. So, but this is the standard nomenclature here. The T equals 7 head structure is all made up of one protein, so it's also a quasi-equivalent structure. So this is the protein which is present at the 5-fold axis and those 6-fold axes. So slightly different structures and interactions with each other, but very, very similar relative to each other. With one big exception, and that's what's down here. This is the portal protein, and it's multiple different subunits. It's actually 12 of them. 12 is also 6 times 2. 6-fold symmetry is very common, where you have the way that you can get the genome out of some of these icosahedral structures. And it turns out this is even true for herpes virus. This is a portal at one of the otherwise 5-fold axes of symmetry. So these are actually not completely symmetric. One of those vertices is a little bit different. So here, portal protein, and we'll see portal proteins again and again when we talk about the different bacteriophage. Turns out these are some of the most conserved proteins. So if you look in databases between a lot of these different proteins, it turns out that the portal protein, which is here in Bacteriophage T4, is pretty similar to the portal protein that you have here, the portal protein that you find in herpes virus. They have pretty similar sequences to each other. And some of the very few that actually have similar sequences across completely different classes of viruses. Also, there's a tail structure. Each of these tail proteins are made up of tails I should say, are made up of 2 different proteins. And then we have 6 of these tail fibers, and we'll take a look at those in a little bit more detail later on. So 6 tail fibers, again, so it's a 6-fold symmetric structure at the end of what's otherwise a 5-fold symmetry axis that you have on your icosahedron. One of the pretty unique things about Bacteriophage T7 is it has this core structure, which you can see really nicely in cryo-electron micrographs that we'll look at in just a second. It turns out that this core structure is really important for getting the genome across the 2 membranes of E. coli, because E. coli is gram-negative, got the 2 membranes and the periplasm with peptidoglycan in the middle. Bacteriophage T4, as we saw last time, has this whole injection where it literally physically injects its tail through those membranes. T7 does this a slightly different way, and it turns out it's basically flipping the core inside out. I think I like to think of this not unlike what happens with a starfish in its stomach, where it does extra organismal digestion in sometimes. So what's present inside the capsid other than the core? It's a 40,000 base pair linear double-stranded DNA genome with unique ends, and we'll see many cases you actually have inverted terminal repeat sequences at ends, but this one was actually kind of different. One of the reasons that people were so excited about this particular bacteriophage, it's actually got a lot smaller genome than this guy, and it's relatively easy to study. You could actually see the genome under the electron microscope, measure how long it was, and then, as we talked about last time for these single-stranded RNA phages, it was a really great way to get a whole bunch of DNA relatively pure that's all exactly the same, or at least extremely similar. What does that genome actually look like? Here it is, our 40 kb genome, and it's really nicely organized, at least in terms of following how it works. We've got early genes at one end and late genes at the other, and then what's called then middle genes here in the middle. Now, these middle genes are almost all what we would originally define as sort of the early genes, and very often people put early and middle genes together. Sometimes people will call these immediate early genes, and then these would be the other early genes. It's all about making the genome. And so making your genome, and then once you've made the genome, then the late genes, again in T7 called the class 3 genes, are about putting the virion together. What do you need to do before making your genome? There are a couple things you need to deal with. One, and we'll also see this in a bunch of other viruses, the most early proteins that you need to express, or sometimes these are the proteins that come in with the genome in the first place, things like the negative strand RNA genomes. This is what you need to shut down host defenses, and then also to make some of the proteins that you haven't brought in with the genome. And it turns out for Bacteriophage T7 and most of these bacteriophage, you can only get the DNA in. Or if you get a protein in, it's going to be a very small protein that's associated with the end of the DNA, because getting through the membranes is really hard. So it's just the DNA which has come in. Well, if just the DNA has come in, you've got to then be making your viral proteins as rapidly as you possibly can. So we'll look at this, keep looking at this as we move along through the rest of this. All of the transcripts that are made from each of these genes have been nicely mapped. There's all these blue lines down here at the bottom, and then these numbers here across the top represent all those GP numbers. Now, if you remember, most of the GP numbers involved in the capsid, like GP10, oops, where is it? Right here. This is one of those late genes. GP10 is the one which is making the capsid, the one that you need huge amounts of, so you also end up with lots and lots of transcripts for that. Another thing which is a little different in terms of this genome is that where you've got these vertical black lines, this is where there's a secondary structure that forms in the messenger RNA, which first gets chopped by a double-stranded RNase, so an endoribonuclease that just cuts double-stranded RNA. and that gives you very stable messenger RNAs. So it turns out that the messenger RNAs that are made here are much more stable than the cellular messenger RNAs. But let's move back a little bit. How do you get that first step? How do you get attachment going on? The receptor for Bacteriophage T7 seems to be the lipopolysaccharide on the outside of E. coli. And so this is not a protein. It's just fatty acids and sugars. Once you have binding to the lipopolysaccharide, there's probably a secondary receptor, which no one's actually found so far. But it, as we'll see, turns out that the virion basically has to walk across the surface of the cell until it finds the right place to insert the rest of its core, which really kind of serves as a drill through both the outer membrane, the peptidoglycan, and the inner membrane. Again, we'll see the animation how that works. What happens once you've drilled your way through the membrane is about 850 base pairs, about 1,000 base pairs get injected inside the cell. Okay, that's great, but it's 40 kb, right? So how are we going to get everything else in there? So this is one of the really unique and fascinating aspects about Bacteriophage T7. Is it turns out that it's the RNA polymerase which pulls the genome out of the capsid and inside the cell. So this is first being pulled in by the cellular transcription machinery and then by the viral transcription machinery. But before we look at that, let's look at how people know about how this interaction was happening and how the virion is sort of walking its way across the surface. So this was a study published 2 years ago, well after the textbook was done, where they showed by looking at cryo-electron tomography that all of the interactions that the virion had with host cells were just single tail fiber proteins. Until you had this big conformational change over here at the end where the core is injected and the DNA is actually released here. Tomography we haven't talked about before, but it's really similar to the cryo-electron microscopy we talked about. We talked about getting higher resolution structures for virions. Cryo-electron microscopy, yeah, quick question?
Yeah, I just had a quick question about the video you showed us last
Yeah.
time about the bacteriophage and the conformational changes
Yeah.
to the cell surface. Is it one mechanism outside the cell surface once it finds a good spot to kind of sit and drill, wherein the tail fibers and the sort of syringe-like area change conformationally, or is this series like it sort of looked like the tail fibers come down, then there's a second conformational change?
Okay, yeah, so the question here, and I'll paraphrase, hopefully I understand it here, is it basically that you have one single conformational change that happens, or there are multiple sequential conformational changes that happen in the infection by Bacteriophage T4? And so that, in fact, was the subject of the paper that that video went with in the first place, was talking about these conformational changes. And so it turns out there are multiple conformational changes, each of which is dependent on the previous one. And so in the case of Bacteriophage T4, you have to have all 6 of these long tailable fiber proteins that associate with the surface. Then you have the conformational change, which pulls the base plate down. Then you have the conformational change, which opens up the base plate, and you get the injection that takes place. So it's multiple conformational changes, each one dependent on the previous one. So, and that's actually not dissimilar to what's going on here. So it's a nice introduction there. But I did want to talk briefly about tomography, because people talk about this, and where does this come from in the first place? But to do that, it's best to, again, return and talk a little bit about cryo-EM. The idea of cryo-EM is you have lots and lots of virions, and you take one picture, and in that one picture, you've got lots of individual virions oriented in different ways. Then in the computer, you take each of those different virions and you average them all together. And you say, okay, this is what any one virion looks like, because we're going to assume they're all the same. And so that's how you do cryo-electron microscopy. Tomography is sort of the flip side of that. Now what you do is you take one virion and you take one picture of it, in one orientation, and then you move it a little bit, change the angle, take another picture. Move it a little bit, take another picture. Move it a little bit, take another picture. And then put all those pictures together with the one particle, and that will reconstruct your three-dimensional structure. Because again, the big problem is you've got 2 dimensions, and how do you go from 2 dimensions to 3 dimensions? So cryo, you assume you've got lots of different orientations for the different one picture that you took of lots of different particles. You average those together, or you take one particle and take pictures from lots of different angles of it, and then put those back together. Tomography is that second one. We take lots of pictures of one particle, and then try and look at each of those. So each of these is going to be reconstructed from one particle and how they actually put them together. So now we'll take a quick look at the movie. This is the animation of this process here, again from that Science article. Okay, let's blow it up and take a look at this. So this is the Bacteriophage T4 infection process. There are a couple of things they did that make this a really impressive study. One of them was taking advantage of the fact that you have so many mutants in T7. This particular mutant has no tail fibers on it. So you're just looking at the individual particle. And that way you can get a really nice reconstruction of this particular particle. So that was the first thing they did. And they could also then really nicely show where this internal core structure is in that single particle. This was done by cryo-electron microscopy, because cryo-electron microscopy gives you much higher resolution. Then if you look at another cryo-electron reconstruction with the tail fibers, in the absence of a cell, you see that they're all piled up at one end. If you do tomography, however, you notice that sometimes you've got one of these tail fibers down, sometimes you've got a different tail fiber down. And so what seems to be happening in this case, and they saw this when they looked at individual cells. Here's that green cell down here at the bottom. Of course, there's a green virus infecting a blue cell, at OMSI anyway. So you have this one interaction that happens here. This is not apparently quite the right place. Probably there needs to be another orientation point here. So now we find another site to bind to with one of these tail fiber proteins. And again, we basically randomly walk around until we find probably a place for this guy to bind to. And then basically we spill our guts through this outer membrane, the peptidoglycan, and the inner membrane, and take this internal core, and it flips its way out through these extra membranes to give you a hole that your DNA can come through out into the outside. And they got all of this from first the cryo-electron microscopy of getting the really nice high-resolution structures, and then use that to interpret their tomography data of looking at the individual particles that were associated with the cell. So that's the process which was used here to figure out that they are literally, walking their way along the cell. And if this is the link to the movie, it's open access, so you can go and take a look at it again if you like. So this is just the cartoon version again of this. What happens? We have capsid. We've now associated with all 6 tail fibers. The core, the otherwise internal core here in gray, is basically turned into a tube, and you release about 1,000 base pairs of DNA from the very left end of the genome. What's present on this DNA? -10, -35 sequences that the bacterial DNA-dependent RNA polymerase binds to. So it will bind right here and transcribe. Transcribing will make messenger RNA. That will get translated by ribosomes, because in bacteria, transcription and translation are coupled. As soon as you make a messenger RNA, as long as it's got a ribosome binding site on it, the ribosomes will bind and start to translate. And this transcription process pulls in about 1/3 to 1/4 of your genome. And this is where all those class 1 genes are. Those class 1 genes are all being transcribed by the bacterial DNA-dependent RNA polymerase. Surprise, surprise, because I'm beating on this whole bacterial DNA-dependent RNA polymerase, one of those genes that's being transcribed and translated is the viral DNA-dependent RNA polymerase, gene product number 1. And we'll see, actually, confusingly, there's some gene products that are less than 1, which is very confusing. But here, T7 RNA polymerase is a single subunit DNA-dependent RNA polymerase. And so this is extremely useful for biotechnology because you don't need to make all these extra subunits and put them back together. And it recognizes a sequence that's completely different than the bacterial promoter sequence in terms of getting binding. So it's about a 17, actually closer to 20 base pair sequence, which the T7 RNA polymerase will recognize, bind to, and then start transcription from there. It's also a lot faster than the bacterial polymerase. It just zooms along the DNA and makes RNA really rapidly. So this is that structure here with the DNA associated with it. Those of you who spend too much time staring at structures of polymerases immediately notice that this looks like a DNA-dependent DNA polymerase because it's got the right hand kind of structure. So fingers, palm, and thumb basically grabbing the DNA, and that's how you're getting the binding to your template. And then the active site is right down here in the base of the palm. So this right-handed structure for DNA-dependent DNA polymerases is true. DNA polymerase 1, DNA polymerase 3, all the eukaryotic DNA polymerases, all of this structure. But this is now an RNA polymerase. So it uses the same template, but is making RNA instead. So that makes all of the middle and late genes. But we also have a couple of other class 1 genes. And again, they're called early genes here, but most of the viruses we'll talk about, this would be people would call immediate early genes. These are the things that are made immediately. So it turns out that there are a couple of genes that come before the T7 RNA polymerase gene. And since you called this 1, you had to call them something less. And so this is 0.3 and 0.7. But these are genes whose products are important in shutting down the host. Two kinds of things you need to shut down. Many bacteria, including E. coli, have restriction endonucleases that will chop up any kind of foreign double-stranded DNA that come inside the cell. And this is a great way of protecting against the double-stranded DNA viruses, most of the bacteriophage. So first thing that happens, the first gene that's made, it actually blocks these. And it also makes sense now, if you think about it, there's only a relatively small amount of DNA which got injected inside the cell after you had this conformational change where the core comes out. Those 850 bases have no binding sites for restriction endonucleases. So you transcribe that DNA. As you're pulling the genome in, you have more and more DNA, some of which has binding sites for restriction endonucleases, but you block the restriction endonuclease by making these proteins. You also inhibit the E. coli RNA polymerase because once you've made these early genes, you don't need it anymore. You've got a the viral RNA polymerase. So you shut down the cellular polymerase because who needs it anymore? We're going to be making all of our viral genes. Yeah?
Well, if the name of the game is to totally expand your population, why not take advantage of there being some other RNA polymerases that can do that for you? Why
Okay, I'm not quite following your question. So
why shut them down if your whole goal is to make as many of your genes as possible?
oh, so why shut down the host RNA polymerase as opposed to letting it do its thing? Well, basically the idea there, and again, this is over-anthropomorphizing the phage, is that, as I mentioned, the viral RNA polymerase is a lot faster, and it is still, however, going to need all of the ribonucleotides, the individual NTPs, to make RNA. If it's now competing with the cellular RNA polymerase, that would be using up this NTP pool, making stuff that the virus could care less about. So the idea here is we're just going to make virus. And so one way of doing that is shutting down the host polymerase. As a completely different start site as well. So it doesn't need the polymerase once it's made these genes right here. And you'll also notice that basically this is just, as long as you've shut these guys down, this is now an enzyme, so it can undergo multiple processes. You don't need very much of this. So once you've got those gene products, forget the cellular polymerase. Yeah?
Can it infect anything besides E. coli if its receptor is LPS?
So turns out that most of these bacteria, the question is, can it infect any other kind of hosts? And the answer is basically no, partly because of this pretty specific requirement for the -10 and -35 sequence from normal E. coli RNA polymerase. And there are, there are some other bacteria that have LPS, but there's a variability in that LPS. And so usually E. coli LPS is pretty specific. But most of these T phages, maybe to back up a little bit, I did mention that they're all infecting E. coli. And so they specifically chose these phage, which are E. coli infecting phage. And most of these bacteriophage are actually pretty specific. They won't infect very many other organisms. Yeah, there's another question over here? Yeah.
Yes. Well, I guess it's not really a question. Like the thing that he was asking about the RNA polymerase, the E. coli one.
Yeah.
It just seems like a really efficient way to shut down the host's immune response to the viral infection, so the host can't, make anything to fight off the infection.
Yeah. So again, it's not if there are other. So another reason you'd want to shut down the host polymerase would be if there's any other defense mechanisms that you need to protect yourself. And we're getting kind of off topic here, but the CRISPRs, so the acquired immune defenses to bacteriophage infection, requires transcription. So if you don't have your own polymerase, you're not going to be transcribing those CRRNAs, which could potentially be helping you. Would be another reason.
Instead of having infectors to specifically knock down specific genes from the host.
Just get rid of that. Get rid of the more stuff you can get rid of from the host, there's always going to be a balance. And again, this is getting a little off topic, but this is going to be true for a lot of these different viruses. There's going to be a balance. You have to have a balance between using as much of the cellular machinery as you can, as far, again, from the virus's point of view, or shutting it down completely. But if you shut it down completely, you've got to bring all of these other proteins with you. They've got to be in your genome. So it's always going to be a balance between these two. And we'll sort of talk about this. The T7 is one of those that's bringing a lot of its own stuff. But when we talk about the single-stranded DNA viruses next week, they're bringing practically nothing. But they're two different strategies. Again, as far as evolution is concerned, if it works, don't mess with it. And so there are lots of solutions, lots of ways to skin the cat, as it were. Sorry for those, feline lovers there. Yeah.
I just had a quick question on the last slide.
Yeah.
So the numbers indicate where they are. How does why does it inhibit the E. coli? How does it work that it inhibits the RNA polymerase before the T7 specific RNA polymerase is created?
Yeah, so the question is, why do you have a gene that inhibits your RNA polymerase before you make the polymerase that you have to have before you've inhibited it?
How do you make it?
Right. So how can you make it before that inhibition? That's because what happens here, this is just the gene. And so as the gene comes inside the cell, it needs to be transcribed and translated. And that delay is long enough to get enough of your T7 RNA polymerase. Yeah?
Does that RNA polymerase only transcribe the viral DNA?
Yeah, so the T7 specific RNA polymerase binds that specific promoter sequence, and that specific promoter sequence is only in front of the viral genes. Although, for biotechnology purposes, what you do is you put it artificially in front of genes, and it ends up being really useful. But it's in a regular, normal, wild-type E. coli. Anytime you've got actually a pretty long sequence, again, about 20 base pairs, you're not going to find those 20 base pairs at random in even a genome as big as E. coli's, which is about 5 million. So that's going to give you really high specificity in terms of binding. Okay, so what are the genes that are made by this T7 specific RNA polymerase? Basically, everything that you need to make more of your own genome and shutting down the E. coli RNA polymerase even more. So it's not just that first gene, the sorry, the 0.7 gene, which does that, also the other ones. Red circles here mean these are the important ones we're going to be talking a lot more about, so you might want to remember them. So particularly here, there's an endonuclease, and this endonuclease chops up the cellular DNA. So again, we're saying in this case, okay, we don't want the cell doing anything. We want to make sure that it's only virus which is being made. This endonuclease chops up the host DNA, but also it cuts the T7 genome at its very end. And that's how you can go from multiple copies of the genome to single copies, which are what gets packaged inside each of the heads in the process. We'll talk more about the lysozyme a little bit later. Lysozyme does kind of like what it sounds, makes holes in the cell wall so the virus can get out once it's done replicating, but it also shuts down the T7 RNA polymerase later on. We'll talk about that. It turns out to be very useful for these biotechnology applications. If we look back at the genome now, here are all of these transcripts which are being made, and it's everything after 1. So 1, this was that T7 RNA polymerase, which now will bind to each of these different promoters. There are lots of them in the T7 genome, and leads to production of very large amounts of RNA for these gene products right here. And you'll notice gene product number 10 right there. Gene product number 10, which is the major capsid protein. So, of course, that's the one that you need the most of. You have, again, all of these different promoters. There are about 10 of them right here. In fact, we know exactly where they are because we know exactly what that sequence is. And you end up, again, with lots of these different transcripts, all of which terminate basically at the same place. So you end up with a bunch of copies here of these genes and a lot less of all of these over here. You do get a little bit of terminator read-through. Termination here happens in a very similar fashion to normal bacterial transcriptional termination. And then I mentioned RNase 3, which chops again at all of these vertical lines and gives you much more stable transcripts. Most messenger RNA in E. coli is pretty unstable and gets broken down pretty fast. On the other hand, in T7, because of this RNase 3 processing and secondary structures at the end, basically blocks the exonucleases. Exonucleases love to chew in at single-stranded pieces of RNA. Here, it's a double-stranded piece, so you don't get as much degradation there. So we've made our class 2 genes, then we go to class 3 genes. We've already beaten on gene protein 10 here, but then GP8, which is that portal protein. And then the last one I wanted to mention here in particular is the holin gene. And it turns out there are many holins you find in lots of different bacteriophage, and holin does basically exactly what you expect a protein with that name to do. It makes holes. It makes holes in the membrane. Lysozyme is really good at breaking down peptidoglycan, but it can't get to the peptidoglycan because it's got to get through the inner membrane. And to do that, you make these holes in the membrane through holins. And in fact, this is a protein that people are really interested in possibly using as antibiotics. It turns out that these holins are really pretty specific for different bacteria and different bacterial membranes. You make holes in bacterial membranes, then those bacteria are not going to be able to survive. And phage have evolved for millions, if not billions of years to make holes in membranes of bacteria. Maybe we can use these as new kinds of antibiotics. Why do you get more of these class 3 genes? Turns out that the promoters are stronger for these genes. How do you get a stronger promoter? The sequence is closer to the consensus sequence, exactly. So that binding site for the T7 RNA polymerase, there's a consensus sequence, and you'll get more of that binding to sequences which are closer to that consensus sequence and less to some of the other ones. Just as a quick mention here, the numbers are not really critical, but we're talking minutes for replication here. So the actual binding to and release of the genome to production of final virus particles is on the order of 20 minutes to half an hour. It's really a pretty amazing process how fast all of this goes. The other thing that happens, as I mentioned before, the T7 lysozyme, as well as breaking down the peptidoglycan, also slows down the production of the T7 RNA polymerase, and that doesn't seem to make a big difference because, these have all been produced, but when you've made the T7 lysozyme, which is one of those late genes, you've already made everything else that you need. These RNAs are stabilized because of that secondary structure. So you just need translation. You don't need any more transcription going on. So the translation is happening there. How do you get replication of the genome? This was another kind of surprise when people were looking at this, and partly how we figured out how replication worked. As we hopefully all know or remember, DNA polymerases have a big problem, and that they always need a primer. What kind of primers are usually used? RNA primers. Where are you going to get your RNA primer from? Well, in this case, it's from the T7 RNA polymerase, because it makes a lot of RNA from very specific positions. But if you have enough T7 lysozyme around, you're going to be blocking the elongation of that polymerase. So you've made your T7 lysozyme, blocking the activity of the polymerase. It'll make these short pieces, but then those serve as the primers for the T7 DNA polymerase. And it turns out that where this happens is at near the left-hand end of the genome, and then you end up with completely normal replication, both strands, leading strand, lagging strand, etc. There is a T7 primase as well, but it turns out that the first step that you need to make is this T7 RNA polymerase. Primase helicase, again, does what normal primases and helicase do. And this process ends up generating, again, the concatamers. You've got multiple copies of the genome stretched out from one end to the other. How does this happen? Well, because you just have one template, right? How are you going to end up with multiple copies at the end? Turns out there are repeated sequences at the end that probably are recombining with each other in order to give you multiple copies of the genome that can then be made into one big, long piece. And then, once you've got this big, long piece with multiple copies of your genome all right next to each other, you need to put these into the head structure. How does that happen? Well, you have one end of the genome that associates with a portal protein. Remember, that's at that normally 5-fold axis, but now is present there with the core structure in it. What you do is this gets inserted. The portal protein has a very strong ATPase activity, basically serves as a pump, and it pumps the DNA inside the head structure. Once it gets inside the head structure and it's filled up in there, that then gets chopped off, and you have a new end. That new end goes and finds a new portal protein. That gets pumped into the next head structure, gets cut off, and so on and so forth. Putting the head together, it's a T equals 7. Again, it's the GP10 protein which is involved in doing this. Making that structure, and particularly making the structure with the portal protein at one end, requires some extra proteins in order to get the full structure to form properly. These are the scaffolding proteins, and those are, again, as we mentioned before, these are classic late proteins that are made. You sort of build the head around these proteins, and then you get rid of them. How you get rid of them, we'll talk about more later when we get through it. And then, of course, holin to make a hole in the inner membrane, lysozyme to break down the peptidoglycan, and poof, now you have lots of virus being released, or virions, I should say, being released. So that's the life cycle of T7. I'll talk about a couple of interesting applications that we have using today, literally, in fact, in my lab, using some of the really neat aspects of mostly the T7 RNA polymerase. And the T7 RNA polymerase is great because, A, it's just a single subunit, so you can make one gene. That one gene will make the one protein that has that activity, and it binds to really specific sequences. So if you engineer now promoters for that T7 RNA polymerase in front of your favorite gene, which is here also known as gene X, that now will be transcribed at very high levels, because again, it's a really fast polymerase, but it'll only be made when you have this T7 RNA polymerase gene around. So this is a really useful technique for making not only large amounts of your favorite protein, but particularly if it's a toxic protein to the cell. If it's a toxic protein to E. coli, then you don't want it to be expressed at all until you're ready to have that being expressed. And so then you'll introduce the T7 RNA polymerase either on a separate plasmid, in some cases even a separate bacteriophage, where you do an infection of this process. Turns out this is also a great way to make RNA, and so a lot of people who are using RNA for ribozymes, for evolution experiments, Dr. Lehman in the chemistry department, who does a lot of origins of life work, he uses, I think he says, bucket loads of these polymerases all the time to make lots of RNA. And then the other T7-like bacteriophage, I mentioned T3 right at the beginning. Turns out T3 is almost identical to T7, except its polymerase has a slightly different recognition sequence. And so now you can have two different ways of making your RNA. I'm not going to talk anymore about GP3. Point 3, just wanted to mention here really briefly that a lot of viruses that are found to infect pathogens, which are now being used for phage therapy, actually look a lot like Bacteriophage T7. This is just one which infects Clostridium perfringens. And I mentioned at the beginning, maybe the most common virus on our planet are these Podoviruses. Because of this particular one here in B, I wanted to mention this in particular because this is work that comes out of Steve Giovannoni's lab just down the road in Corvallis. They discovered this organism, which they call SAR11 for Sargasso C11, probably the most common bacterium in the oceans. People thought for a long time the reason it was so common is because no viruses infected it. Well, you can see here that actually there are lots of viruses that infect it, and because it's the most common bacterium in the oceans, and these are some of the viruses which infect it, it's not surprising that this guy would be one of the most common viruses that are out there. And in fact, when people look at sequences present in databases, that's exactly what they see. You go out in the oceans, you sequence all of those little dots that we saw in the first lecture. You find sequences very similar to this sequence right here, which again makes a lot of sense. And I just want to finish up with this really amazing cryo-electron microscopy reconstruction of not Bacteriophage T7, although it's really similar here, where you can see the nice five-fold axis of symmetry. It's hard to see here, but that's 5, 6, 6, 5. So again, nice T equals 7 icosahedrally symmetric particle. There's the core structure on the outside with these tail fibers up against the virus particles, not associated with cells yet. This core protein here in the middle. But much more importantly here, and that's one thing I really like about this particular cryo-EM structure, you can see the DNA. All of these black lines in here are the DNA, and you can see how amazingly tightly packed it is inside this actual structure. And so with cryo, if you've got a ordered structure, and again, these single particle reconstructions, you've looked at literally thousands of these things, all of the DNA is at exactly the same place in each one of these particles. Otherwise, you would never see it. So that's it. Cool stuff you can do with synthetic biology and evolution with these guys, but kind of a side point. Have a good weekend. We'll see you on Monday.